Hello, I'm Professor Matthew Schmidt, and this is Genetics. In this session, we're going to discuss, in our general exploration of DNA structure and replication, the structure of DNA. If you're following along in order, last time we spent a bit of effort going through the experiments and the rationale of determining, in fact, that DNA was the genetic material. This, we had said, was pretty much confirmed by about 1952, and then people got very interested in DNA, and you probably know that this ultimately culminated in Watson and Crick's Nobel Prize winning discovery of the exact structure of DNA. It's very important to remember in all of biology and even in other things that structure and function are so intimately related. So in this case, we understood the function of the gene of DNA much more than we understood the structure to begin with, but the structure, in other words, they're not separable. We mentioned certain characteristics that a molecule would have to have in order to be the genetic material, and the structure of DNA that Watson and Crick ultimately elucidated explains how this wonderful molecule can do that job. So before we get ahead of ourselves, the last question that we asked when we were talking about it last time was, what was biochemically known about DNA? Meaning, at the time in the early 1950s, right before Watson and Crick, I had mentioned to you that biochemists were aware of the existence of DNA and knew certain things about its general structure. It was known to be present in chromosomes, but it was thought to be a relatively unimportant just a molecule that was part of the fabric of the chromosomes, not having to do with information. So what was in fact known at that time was that DNA, like so many biological molecules, was a polymer, which means that there are subunits that are strung together into, in this case, a very long line. The subunits are known as nucleotides in this case, and there are 4 types of nucleotides, which we're about to get into. Just remember, originally it was thought that protein was the genetic material. This is not the only reason, but it was in large part because there are 20 different amino acids from which to create variation. We only have 4 here, but it turns out that you really don't need a lot of different types of subunits to encode a ridiculously large amount of information. So there are 4 types, and nucleotides, as monomers or subunits go, are relatively complex. So each nucleotide has a sugar, and in the case of DNA, that sugar is called deoxyribose. It's a 5-carbon sugar. There's a phosphate group, and there's also a nitrogen-containing base. So let's look in a little bit more detail at this. So we did say that there are 4 different bases, but my point here is that it is the base and the base alone that causes one nucleotide to differ from another. In other words, all 4 of the different nucleotides are the same in the sugar and the phosphate group part. They differ only in the base part. So 4 different bases allow us to have 4 different types of nucleotides. Those 4 are divided into 2 categories based on the size of the base. So purines, so-called, purine bases contain 2 rings. I'll show you a picture in a moment. And those bases are adenine and guanine, often abbreviated as A and G. The pyrimidine bases only have 1 ring. They're literally shorter in length, which is important in the overall structure. Pyrimidines have 1 ring, and those are cytosine and thymine. And as you may know, those are often abbreviated as C and T. A lot of times when you're dealing with DNA sequences, we want to just write it as C-A-T-T. We have to know what we're talking about, but a lot of times the abbreviations are used. However, for right now, we want to go into a little bit more detail because the base itself is named either adenine or guanine, cytosine, and thymine. But when the base gets attached to the sugar, first of all, this is how they're built, that's called a nucleoside. And then when the phosphate group is added on, that's when they become a nucleotide. Many of you may know this, but I know I didn't know it for a long time when I was a student. The names change when the base is incorporated into a nucleoside or a nucleotide, and the names change in the following way. So a nucleotide containing the base adenine is now referred to as adenosine something, which we'll get to in a minute. And it's a good idea to, I was going to say memorize, I'm not positive if that's the word, but it's certainly good to recognize if you see adenosine, you know that they're talking not just about the base. The base is always adenine. Well, in that case, if the base is guanine, the nucleotide built, or I should say that contains that base, is going to be guanosine. So those two, adenine and guanine, they change in a similar way to, instead of nine, they change to ozine. The cytosine and thymine change in a little bit of a different way. Cytosine becomes cytidine if it's in a nucleotide, and thymine becomes thymidine. Now, I want to show you pictures of these and talk a little bit more about the naming conventions. But before we leave this slide, I think it's very important to, a lot of people ask, how can I easily remember which are the purine bases and which are the pyrimidine bases? And it turns out there are a couple of good ways to remember. If you think of one on your own that works, that's great, but I have two for you. One of them is that the word pyrimidine has a Y in it, right? And both the bases cytosine and thymine also have Y's in them. You'll note that none of either the word purine or the purine bases have a Y. So some people like to think if I see Y, that means the Y's go together in a sense. Cytosine, thymine, pyrimidine. Another way that focuses more on the purines is to remember this saying. It's a little silly, but a lot of these sayings are. Pure as gold. If you think of gold being a pure metal, pure as gold refers to not only the P in the purine, but the word pure is in purine, as for adenine, and gold for guanine. So both of these are going around in my head. You can use both of them. You can pick one, or as I said, you can make one up yourself, but you're not going to get better than this. I think you are expected to know which are the purines and which are the pyrimidines. So figure out, use one of these ways or both. Now, let's look at some excellent pictures of the full nucleotides, talk a little bit about it. Now, first of all, so often the nucleotide is depicted this way, but you'll notice that there's one phosphate group here attached. So these are monophosphate versions of the nucleotides. It turns out that there can be, I'm not going to draw out the whole phosphate group here, but I'm just going to write PO4 to mean a phosphate group. There can be two of them stuck on, in which case this is a diphosphate version, and there can be even three of them stuck on. The chemistry isn't exactly correct, but then you would have, with three, right, a triphosphate situation. So nucleotides may be monophosphate, diphosphate, or triphosphate. When they're in the pool of nucleotides in the cell, they tend to exist as the triphosphate version, and when they get incorporated into the chain, two of the phosphates are used in that incorporation. They're used up. So in the chain, they exist as we've drawn it here as a monophosphate nucleotide. So let's look at the naming conventions here. So the base adenine is right here. You can see that there are two rings there. It's a purine, right? Also, this is it's biochemistry, but it's not a bad idea to remember that the base is always attached to the 1' carbon of the sugar, whereas the phosphate group is always attached to the 5'